Difference between inanimate objects and living things. The main differences between inanimate objects and living things are as follows. Life. 1. Inanimate objects. Inanimate objects are not alive. 1. They do not generate their own energy and do not grow or reproduce. 1. Examples of inanimate objects include rocks, metals, water, and stones. 2. Living things. Living things are alive. 2. They grow and reproduce, and most of them obtain energy through metabolic processes. 2. Examples of living things include plants, animals, and microorganisms. Cell composition. 1. Inanimate objects. Inanimate objects do not have a cellular composition. 1. Most inanimate objects do not have a cellular structure. 2. Living things. Living things are composed of one or more cells. 2. Cells are the basic structural unit of living things and perform metabolic activities and biological functions. 3. Metabolism and response. 1. Inanimate objects. Inanimate objects do not perform metabolic activities and have limited responses to environmental conditions. 1. Chemical reactions are determined by environmental factors. 2. Living things. Living things have the ability to utilize chemical energy through metabolic activities and to respond to the environment. 2. Living things can adapt and respond to the environment. 4. Growth and reproduction. 1. Inanimate objects. Inanimate objects do not grow or reproduce. 1. Inanimate objects can only change through chemical or physical changes. 2. Living things. Living things grow and reproduce. 2. Living things reproduce by giving birth to offspring or producing seeds. 5. Evolution. 1. Inanimate objects. Inanimate objects do not evolve. 1. Inanimate objects do not undergo their own evolutionary process even if their chemical or physical properties change. 2. Living things. Living things can diversify their species and improve their adaptability through the process of evolution. 2. The evolution of living things is driven by genetic changes and natural selection. Summary. Inanimate objects are not alive, have no cellular composition, do not perform metabolic activities, and do not grow or reproduce. Living things, on the other hand, are composed of cells, obtain energy through metabolic activities, grow, reproduce, and evolve. Viruses. A special type of microorganism. Viruses are a unique type of microorganism that falls somewhere between living and non-living things. They have the following key characteristics. Structure. 1. Viruses have a very simple structure. 1. They are typically composed of genetic material, usually DNA or RNA, surrounded by a protein capsid, coat. Life status. 1. Viruses are not living organisms and do not perform metabolic activities on their own. 1. This means that viruses do not produce energy or grow. Reproduction. 1. Viruses require a host organism's cells to reproduce. 1. Viruses infect host cells and replicate their genetic material, producing new virus particles. 1. This is the way viruses reproduce, using the host organism's cells to multiply. Disease and infection. 1. Viruses are a major cause of many diseases. 1. Viruses can infect the cells of a host organism and cause illness, making viruses medically important. 5. Evolution. 1. Viruses evolve. 1. The genetic material of viruses can change over time, leading to the formation of new virus variants. 1. This is the result of viruses interacting with their hosts and being under evolutionary pressure. Viruses are microorganisms, but they differ from typical living organisms in their characteristics. They use host cells to reproduce and cause diseases, and these characteristics make them one of the medically important entities. Gene editing technology. Gene editing technology is the ability to precisely cut specific DNA sequences. It can be used to correct genes, insert new genes, or remove genes. Gene editing technology can be divided into two main categories: CRISPR Cas9 and TALEN. The CRISPR Cas9 system is a system found in the immune system of bacteria. The Cas9 protein binds to a specific DNA sequence, and the CRISPR RNA guides the Cas9 protein to that sequence. The SOS9 protein cuts the sequence, and the cell repairs the damaged DNA by correcting the gene or inserting a new gene. The TALEN system is a technology that uses TALEN proteins to cut DNA. TALEN proteins bind to specific DNA sequences and cut DNA. The TALEN system is less accurate than the CRISPR SOS9 system, but it can be used to cut a wider range of DNA sequences. Gene editing technology has a wide range of potential applications. In the medical field, it can be used to treat genetic diseases or develop new drugs. In the agricultural field, it can be used to improve crop yields or develop new varieties. In the environmental field, it can be used to reduce environmental pollution or develop new energy sources. Gene editing technology is still in its early stages of development, but it has the potential to revolutionize many fields. Here are the main application areas of gene editing technology. 1. Medical field, treatment of genetic diseases, development of new drugs. 2. Agricultural field, improvement of crop yields, development of new varieties. 3. Environmental field, reduction of environmental pollution, development of new energy sources. Gene editing technology is still in its early stages of development, but it has the potential to revolutionize many fields.